Good morning, dear students. Today we are going to see about the sauce repair. Have you heard about this sauce anywhere? I think you heard about sauce in tomato sauce and some other sauce. Anyway, that doesn't mean here. It means a different method of repair. And what is the expansion of this word sauce? It means save our soul. That means that this method of repair need to function in some emergency or in some severe DNA damage conditions only. Okay, that's what written here. The source repair that begin to act when the DNA got severely damaged or its damage means damage reach a limit. I mean, or it crosses a limit of damage. That time, the source method of repair begin to act, and the source response or source repair, it is existing in bacteria or prokaryotes. The source type of repair is not at all reported in any eukaryotes so far. But that doesn't mean that eukaryote cannot repair severe DNA damage. There present some different mechanisms. Okay, those mechanisms we are not going to learn in this section. We are only learning the source response, source method of repair existing in bacteria. The source response has an operon for the source genes. So here also some genes are there. That genes are responsible to make this source response. And then what do you mean by this operons? Operons means it is a cluster of functionally similar genes. And all these genes sometimes will produce a single mRNA, and from this different proteins that are having similar functions are producing. And anyhow, all these functionally similar genes are controlled by a single promoter and an operator in the beginning. So such a cluster is called operon. And most of the operons are having a regulatory activity. Regulatory activity on other genes. So here also this source response is not at all any repair mechanism. It only producing certain proteins or enzymes that is helping the cells to overcome the severe DNA damage. Okay, clear? So source response is not at all doing any repair to DNA. Instead, it is producing some proteins that are helping to repair the severe DNA damage. Clear? So let's see the process. Suppose this is a DNA, and as it is exposed to some UV irradiation, If it is a minor type radiation, our normal DNA repair systems can correct that one. For example, photoreactivation can correct this type of minor levels of DNA damage because of UV radiations. We have already learned in our prior videos about what is photoreactivation. So I am not going to again rewind the entire process here. Leave that. But if the DNA is exposed to too much UV radiation and got damaged severely, then the available DNA repair system in the cell cannot repair that damage. Then what can do? Anyway, need to overcome. Anyway, need to do the DNA replication for this DNA or the damaged DNA also, but it is not possible as that damage or that lesion existed in that DNA. That time, the only solution is to have a bypass, means instead of directly going to any other type of repair mechanism, yeah, that the repair system, it switched to another pathway, that is source repair. When it switched to that pathway, when the damage is severe, at that time it switched to source repair. So let's see that. So this is that operon I already explained to you. Promoter is there, then operator. It is masked by some other protein. I will explain to you what is its name later. Promoter, operator, then a set of genes downstream. And as the DNA got damaged, the RNA pole came to transcribe these genes. To get the proteins that need to do the DNA repair. So that RNA pole came, but it cannot proceed downstream. Why? Because its path is blocked. It is blocked by another protein called LEX-A. This is the LEX-A. This LEX-A is repressing the transcription of this operon. LEX-A is a repressing protein. It prevent the transcription of this operon. And the DNA got severely damaged. At that situation, there come another protein called the REC-A, and this REC-A stimulates the autocatalytic catalytic activity of LEX-A. Means LEX-A by its own can cleave it and can move away from that operator. So let's see that. REC-A is moving to LEX-A and simulating its autocatalytic activity, and by that way LEX-A is removed or it moves apart from the operator. Then the RNA pole can transcribe that downstream genes. And from first UVRA gene form an enzyme, and it is an important chief enzyme in nucleotide excision repair pathway, that is UVRABC endonuclease. The name of this enzyme is UVRABC endonuclease. So it tried to correct that damage, but sometimes this UVR ABC endonuclease failed to repair that DNA damage. It fails. So then what can do next? Then the other two gene, its name are UMUC and UMUD. From these two genes form two other proteins. UMUC protein, this is a UMUC protein, and this one is UMUD protein. These two proteins are formed from these two genes, and they combine together to form a polymerase, that is DNA polymerase 5. We know that DNA polymerase is the enzyme that is doing DNA replication. DNA replication. Okay. So, the situation that we have seen here is that from the UVRA, they are form an enzyme UVR-ABC endonuclease, and that too failed to correct the repair, means that damage. Then anyhow need to replicate this DNA, means damaged DNA, not this DNA, that damaged DNA. So, for that, error-prone DNA replication. Listen, that term, error-prone. That means that with all the damages, that damaged DNA need to undergo replication. So, for that, this enzyme is helpful. So, there is a name, a term for that error-prone DNA replication. That means by incorporating errors, that DNA is undergoing replication. It is called a translation DNA synthesis. That is why those cells that are exposed to too much UV radiation, it is giving the same effect to its DNA. It is getting severely damaged, and the cell cannot repair that damage, and it is undergoing replication, and then that cell is becoming a mutant cell. So that's why UV radiation, exposure to too much UV radiations are causing mutations. Okay. So that is source repair. Source repair is helping the cell to overcome the damage. If it is not possible, then it is supporting that cell's DNA to complete the DNA replication even with the errors. So that is source response or source repair that is present in prokaryotes like bacteria. I think it's very clear to you. Thank you.
I'm an icon, man. My girl about a 10. I should fuck her in a Benz.